So continuing on with our digestive system, we've made it now all the way down to the stomach. Our material that we brought in, that was a bolus, and now in the stomach, what do we call it? Chyme, because we've added enzymes, we've added hydrochloric acid. So we are going to mix and churn the chyme. Now we began to see peristalsis in the esophagus. These muscular wave-like contractions moving through. We're really going to see it in the intestines, especially where we have the circular muscle layer and the longitudinal. It's usually the inner circular, the outer longitudinal. That's our generalized plan. But you see in the stomach we're adding one. Which one are we adding? Oblique. What does oblique mean? Yeah, to me it's not straight. You know, because the circular and longitudinal are 90 degrees. They're perpendicular to one another. Oblique is at an angle. And so this oblique muscular layer is going to allow you to really churn and mix the material. Otherwise, you'll just be compressing, shortening, compressing, shortening. Now you're just really mixing it up. So a lot of mechanical digestion, a lot of chemical digestion is occurring in the stomach. Now in the stomach, we saw that extra muscle layer for the mechanical. Now we're going to get into all the chemical digestion that occurs from a lot of different cells that are present in the mucosa of the stomach. And what we're going to see is that once you get into the lining of the stomach, if you could be in the stomach and look at the wall, it's going to look like Swiss cheese. You're going to see all these holes. And these holes are going to open to a gastric pit. And you can see that extending down into the submucosa. And these cells are going to be continuous on the surface down this gastric pit. And as this extends downward, this is going to branch, and this is going to be a tubular gland. And all of the cells down at the base of this area, this is going to be our gastric glands. And these gastric glands and the pits and even the lining, they're going to have these different cell types that are going to help us with the process of digestion, as well as producing materials so that these hydrochloric acid and enzymes do not destroy the stomach itself. That's kind of important, right? You don't want to digest yourself every time you eat something. Food possibly can go in here, but you understand you're making a lot of material that's being pushed upwards too. So if things get in here, it's being cleaned out by all the secretions from these cells coming through. Now, since we're going down toward the submucosa, we do have this muscularis layer, muscularis mucosa, not the big muscle layer, but this is that little strip of muscle. And this smooth muscle is going to help get those secretions, kind of squeeze them from the base and help them go upwards. So not only do you have the pressure of the secretions themselves, the muscle is going to help express it up into the lumen of the stomach. So collectively, everything the stomach lining makes is going to be called gastric juice. And we're also going to have hormones, cells that are putting hormones into this gastric juice as well. And we're going to start by looking at the surface cells and some of these cells that extend down into the very beginning, which we call the neck of our gastric pit. And so these are surface mucus cells, the lining of the stomach. Sometimes people call these surface lining cells or SLCs. And really the job of these cells is protect you from hydrochloric acid. Because as the name says, they're producing mucus. But this mucus is going to be very alkaline, and it's going to be alkaline to offset the hydrochloric acid that's in the stomach to prevent that low pH material from damaging the lining cells themselves. So you're going to neutralize the hydrochloric acid right at the surface of these cells. This material is not being made so much that it's going to diffuse into the lumen of the stomach and make that an alkaline environment there. This is kept right on the surface to just protect the lining. Because you've got to have this low pH in there to help process the chyme that you've just ingested. Now we're going to extend our way down into the gastric pit, and this is where we have what are called mucous neck cells. Now these aren't necessarily exactly the same mucus producing cells as we had on the previous slide we have at the surface, because these mucous neck cells, while they do still produce mucin, and that's the component that when hydrated makes mucus, notice they're producing acidic mucus. And basically what's happening is you've got to keep the glandular area down here more acidic. And the cells on the surface are the ones that can't stand the low pH. So when you've got all of this mucus with the bicarbonate in it to buffer, you want to keep that from getting too far down. Otherwise, you're going to interfere with the production of hydrochloric acid and its release, because you would neutralize it down here. So these mucous neck cells are different than the mucous surface lining cells that are buffered against the pH. But notice both, they're helping lubricate the stomach. Because you got a lot of material in there, there's a lot of grinding and churning going on. You don't want abrasion to happen. So the mucus is lubricating the lining of your stomach. So both, in fact, do that regardless of their effect on pH. Now the parietal cells, I almost think of these as the one of the two business end of our chemical digestion, because the parietal cells, they're going to add two things. This is one that is least thought of as far as the function of the parietal cell. Intrinsic factor. This is a glycoprotein that is produced, and it's important to have this for the absorption of vitamin B12 in your diet. If you don't have intrinsic factor, you're not going to be able to effectively absorb B12, and if you don't have B12, you're going to be anemic. And there's a condition that people are unable to produce intrinsic factor, and I believe that condition is called pernicious anemia. Now, this is important, don't get me wrong, but the other substance that parietal cells make is absolutely critical because they produce the HCl, the hydrochloric acid. Now, when we think of this cell and the other cell that's going to be producing enzymes, you know how we had the serous glands and the mucus glands? The mucus glands produce, of course, mucus. The serous glands are producing what? Serum-like, which has enzymes and proteins. Okay? Another way to describe what's going on here, cells that produce acids, and in this case hydrochloric acids, are referred to as oxyntic. Oxy, O-X-Y, oxyntic. Versus the cells that produce the proteins and the enzymes, those being the chief cells, those are coming next, those are called zymogenic, zymo for enzyme. So parietal cells produce intrinsic factor and the acid. Now, is it any surprise that the hydrochloric acid is a good antimicrobial? Yeah, any nasty that is on your food and gets into your stomach, it's going to be destroyed primarily by the low pH environment, the hydrochloric acid. So it helps to denature proteins and carbohydrates and all the stuff in our food, but it will also help prevent you from getting sick. Now, we also eat, we're omnivores, right? Do we know what omnivores mean? We can eat anything. So we're not just carnivores and meat eaters, although I like my meat. We can also eat plant material. And plant has the cellulose, and we don't have enzymes to break down cellulose. But the hydrochloric acid helps to break down those cell walls from plants and bacteria, as well as the connective tissue that's in a lot of our meat. So hydrochloric acid is doing more than just denaturing protein. It's helping us with microbes. It's helping us break down the plant materials. And then one of the cool functions of hydrochloric acid, and we've already seen this when we look at the blood clot cascade and we look at complement proteins in the immune system. What happens when we see O-gen at the end of a word? It's inactive. We've got to get the O-gen off to activate this enzyme. So we have an important enzyme that's going to aid in digestion called pepsin, but it starts off as pepsinogen, and hydrochloric acid is going to help clip off the O-gen, and pepsin, which is resistant to acidic environments, it's going to be active. It can help us with digestion. So hydrochloric acid is doing a lot more than just beat nasty low pH environment, just to denature proteins. And we have other acidic enzymes, lipases, and we know what lipases are, right? What do they eat? They eat lipids. They eat lipids. Lipases break down fats. All right, so here are our chief cells. Chief cells produce pepsinogen, again, one of those digestive enzymes. And so since chief cells produce proteins and enzymes, this is when I was talking about zymogenic is the type of cell that we find with the chief cells. Oh, one more. Not just, this enzyme, it produces quite a few, but there's the gastric lipase. And often when you see an enzyme with an organ in front of it, there's going to be other organs that are going to produce its own flavor of a particular enzyme. So we start fat digestion in the stomach, but we're only doing 10 to 15%. We got a long way to go before we finish our fat digestion. Now, here is a cell type probably you would be least familiar with, although we did talk about the endocrine system. We have individual cells, and some of these cells are called G cells. I think there's a lot of cells with these letters. But these are a type of a cell called an enteroendocrine cell. Entero or enteritis is usually an infection of what? Yeah, I mean, it's going to be your gastrointestinal tract. So enteric nervous system, entero refers to your digestive tract. And so these are individualized endocrine cells in the stomach that produce hormones. And in this case, this particular type of enteroendocrine cell is called G, because guess what it produces? Gastrin. And so these G cells produce gastrin that is going to cause the stomach to start churning and cause the gastric glands to make their secretions. And we're going to see there's a feedback mechanism that gets this started. We'll see that a little bit later down the road. So here's our synopsis picture of our 5 major players in the surface lining, the gastric pit, and down into the gastric glands. These are the business end of our chemical digestion. We know that our stomach is mobile, right? We know our stomach moves food. It's doing mechanical digestion. We can often hear it doing that, right? So with our stomach, we're going to see that we're going to take the bolus, and where do we have the bolus? Starts in the mouth, down the esophagus. When that plops into the stomach, now we're going to start churning it, mixing it. We've got all our gastric juice. That's where we make the chyme. And then after our stomach processes it, we're going to pass through the pyloric sphincter. Remember, this is the pylorus of the stomach. That's why we got that name, pyloric sphincter. And into the small intestine, and the first part is called the duodenum. So we've already said the 3 layers of our muscularis, the inner oblique, the middle circular, and the outer longitudinal. we're mixing and churning. So the mechanical digestion. The mechanical digestion is reducing the size of the material, breaking it up. What is that going to do for you physiologically?
Allows it to go into smaller regions and be absorbed.
It's in smaller places, but we're not ready to absorb yet. More surface area for the acid and the enzymes to work on. If you're just going to have an intact Big Mac sitting in your stomach, it's going to take it a lot longer to process than the one you chew up into small bits and swallow, and your stomach makes it smaller and continually smaller and smaller and churning. All right? Now, once we churn and mix and we have our chyme, we're eventually going to need to allow that material to move into the duodenum. So we're going to see that because of gravity and because of muscular contractions that are going on, we're going to have a pressure gradient. And that pressure gradient is going to be lowest up by the dome. What do we call this part of the stomach? That dome shape up here? It's no fun. Fundus. Fundus. It's no fun to have a hunchback, right? So the fundus is going to have the lowest pressure and we're going to have the highest pressure and the lowest pressure down here as you're squeezing down. It's like squeezing toothpaste out of that tube. And so that's going to move our chyme down into the pylorus of the stomach, and that's where we encounter the pyloric sphincter. Now, when you have food in your stomach, what condition is the pyloric sphincter in? It is closed. You're processing the material. So we're going to get signals depending on how long material stays in your stomach. How long can food stay in your stomach?
6 hours.
2 to 6 hours. 2 to 6 hours, depending on what the meal is. You know, if you eat a steak, it's going to stay in there longer than Jell-O. So, now, as materials leave the stomach and move into the duodenum, the pyloric sphincter just doesn't open wide and everything just slide down all at once. The pyloric sphincter is going to open, allow a little bit of this to move in, and then it's going to close again. Now, retropulsion is sort of, I don't know if that's kind of a new-ish kind of term. The term I think about is regurgitation. We all know what regurgitation is, right? Yeah, throwing up, going backwards. You do not want the chyme, once it gets to the duodenum, to go backwards. If it just would move back and forth, you're not making any progress. So you're going to push the material through, the gates are going to close, and then the duodenum and peristalsis can move it on down the line. But you do not want it to go backwards. Regulation, you can already tell with the peristalsis, with the sphincters that have to open and close, with the hormones and the release of certain secretions. There's got to be a lot of regulation here. So as we look at what's going on in the stomach, we're going to see, much like with swallowing, you remember we had our phases, voluntary phase, involuntary, and then we get down to the stomach. Well, we've got 3 phases here of your stomach and the regulation of its digestion, the cephalic, the gastric, and intestinal. So the first phase, cephalic. I'm hungry. That chocolate cake looks good. All right? These nerve signals are going to say, I want some food. Feed me. And so we're going to have this going. Cerebrum. Cerebrum's going to say, I'm hungry. Hypothalamus. Hypothalamus is going to say, hey, medulla, you better get the stomach ready. Something's coming. And then we're going to start having this vagal. Okay, what does vagal mean? You ever heard vagal before? The vagus nerve, exactly. So sympathetic. Well, and in this case, we're talking about digestion, so it's parasympathetic. So we're going to start stimulating the stomach. Have you ever been thinking about something to eat, or you see a commercial on television, and there's something really good, like that Arby's brown sugar bacon sandwich thing? You see that, and you're thinking, oh, that looks so good, and your stomach growl. That's what's going on, because your eyes are going to be directly related to your stomach in that case. So we made it from the cephalic cephalic is head, brain, so this nervous system stimulation. Now the stomach is going to take over, and we have baroreceptors, and they're detecting what? Pressure. And we've also got chemoreceptors that are detecting, okay, what are our enzymes and what is the acid doing in our stomach? And all of this, again, the medulla and that, not digestion center, I forget exactly what the name is in the medulla, but that is controlling what is going on through our parasympathetic nervous system. And so once we get these signals, the medulla is going to say, guess what? Muscles, let's get going. Motility, let's secrete more material. And then when we've got food down in the stomach, we've not just thought about it, we've actually eaten it. Now we're going to get gastrin. Release. What cells make gastrin? This is an easy one. G cells. G cells. Calcitonin. What cells make calcitonin? C cells. See, so these are some of the easy ones. So in this case, gastrin is going to be released, and that is going to cause almost a positive feedback because gastrin is going to cause these contractions. And these contractions are going to help express the hydrochloric acid out of the gastric glands and into the lumen of the stomach where the food lives. So now, made it through the stomach. We saw the food, we ate the food, we've crunched up the food, we've got our chyme ready to go, moved into the duodenum, which we now call the intestinal phase. We're going to get to the segments of the small intestine, but the very first section is called the duodenum. And so we're going to detect what's going on in the duodenum. Because the duodenum, the cells that line the duodenum, they are not protected from acid. And so it's important for us to know, okay, what's happening with the chyme here, because the duodenum has to be taken care of. And we're going to see how the duodenum does it. And so these chemoreceptors, they are going to cause the release of cholecystokinin, which is abbreviated CCK, and secretin. And we're going to talk about these more as these really regulatory hormones and factors and where they're made and what they do. But cholecystokinin is going to say, okay, you know what? Stomach, you can slow down. We've got it in the intestine now. Secretin, yeah, stomach, slow your roll. You don't have to be producing as much hydrochloric acid because we're taking over now. So do you see how it's a way to shut down the stomach now that the chyme has moved into the intestine? And so by shutting down the hydrochloric acid, that's one way we're protecting the duodenum. We're not going to have too much acid. But we are going to see when we get to the duodenum, there's a special gland, it's in the submucosa, that produces a bicarbonate-rich secretion that's injected into the duodenum to neutralize any acid that might be making it there. It's called Brunner's glands, but there's a slide coming up that will show that. Now, as we get into this intestinal phase, we're going to be feeding back information to the medulla, that digestion center. And so we're going to see that there are decreased signals going to the medulla, and the medulla is like, okay, we're not feeling the pressure. We're not sensing the chemicals, and so that's going to start our parasympathetic signaling to the stomach to say, you know what? We're going to inhibit the vagus, which is going to stop signaling that gastric phase. So once we get into the intestine, the feedback that's coming to the medulla is going to inhibit what's going on in the stomach and shut that down because there's not much left to do in the stomach. We've already processed that material. All right, so food stays in the stomach 2 to 6 hours. Once we get in the small intestine, we may be there a while. 12 hours. Now, if you were asked, what is the primary function of the stomach? What would you say? It would be tough for me to know whether it's mechanical or chemical, but digestion as a whole. What is our primary function of the small intestine? Absorption. Now, we're still going to do a little bit of processing of chemical digestion in the duodenum. We're going to mechanically process as we undergo peristalsis and move this toward the colon. But the principal primary function of the intestine is going to be absorption. And look, most nutrients, water, electrolytes, vitamins, everything. And the small intestine in order is the duodenum, the middle segment is the jejunum, and the last segment before you get to the colon is called the ileum. Those are the three segments. Now, we've already seen we're going to get that chyme from the stomach, from the pylorus of the stomach into the duodenum after it passes through the pyloric sphincter. Looking from the outside, you would be very hard-pressed to tell what's duodenum and what's jejunum because they're continuous. But on the inside and looking at the histology, there's going to be some telltale signs if you're going to see histology in a lab. The other way you can distinguish the duodenum from jejunum is the duodenum is retroperitoneal. Almost all of the duodenum is retroperitoneal. And it's also in the duodenum when we're still doing chemical digestion. We're going to get a lot of secretions that are coming from the liver where it's produced, stored in the gallbladder, and that's going to empty into the duodenum. What material do we get from the gallbladder? Bile. The pancreas is going to make a ton of enzymes. We're going to have pancreatic amylase, pancreatic lipase. See how we changed the name from gastric to pancreatic? So all of these secretions are going to be working on the chyme still in the duodenum. And then here's the way histologically you can tell if you're looking at a section of small intestine which segment it is. Because here in the submucosa, you have this huge assortment of acini and glands. These are submucosal glands or Brunner's glands that produce that alkaline secretion to neutralize the acid in the duodenum as soon as that acidic chyme hits. And that way we're going to protect not only the lining of our intestinal tract, but we're also protecting all of the secretions coming from the gallbladder and the pancreas. Because the enzymes coming from the pancreas are not meant to be active in acidic conditions. That acid from the stomach would neutralize all of these enzymes, and so we have to neutralize every bit of that acid, and that's what the Brunner's glands do. All right, the jejunum. This one is fairly innocuous. It's the middle region. Okay? You're still processing the chemical digestion from the pancreas and the liver. You're still absorbing, and it's intraperitoneal. There's not much else to say. Histologically, the jejunum is nothing. That may sound weird. And I really can't define that any better until we look at the ileum. So let's just skip to the ileum. This is the last region. It's going to terminate where you join the small intestine to the first part of the large intestine, which is called the cecum. A cecum is a blind-ended pocket of your intestines. And so that junction is going to have a sphincter to control the passage of chyme from the small intestine to the colon, the large intestine. It is called the ileocecal, sometimes people call it the ileocolic for colon, the ileocecal valve. Now, in a histological slide, you're not going to see those glands, the Brunner's glands down here, but you are going to see these large lymph nodules. And hopefully you remember those from the lymphatic system. Those are Peyer's patches. So if you see Peyer's patches, where are you in the small intestine? The ileum. If you see Brunner's glands, where are you? Duodenum, the duodenum. If it's just connective tissue and there's nothing here, What is it? Jejunum. See, that jejunum is process of elimination. See, Pyrus patch, okay, that's going to be ileum. Oh, look at those Brunner's glands. That's got to be duodenum. Well, there's nothing here but connective tissue. Nothing, then it's jejunum. And that's how you distinguish those three regions, looking at the histology. Maybe y'all will have those in lab. Yeah, we're continuing absorption in the small intestine. So really, the small intestine, as I mentioned, little bit of chemical digestion in the duodenum. But as you move toward the ileum, that chemical digestion is completing itself. But you're going to do absorption in all the duodenum, jejunum, and in the ileum. As we look at the lining now and the lumen of our intestinal tract, we're going to see some specializations. Because if we want to absorb every bit of the good stuff we're eating and drinking, what is one way we can ensure to optimize absorption? By increasing what? Surface area. So everything we're about to talk about, these specializations for increasing surface area, other than just the long tube itself, we're going to collectively put these together to increase the surface area in the intestines by 600-fold. Not 6, not 60, 600-fold. And if we're increasing in surface area that much, you can bet everything you eat is going to get absorbed. Especially if you eat right before you go to bed. Because that's when parasympathetic is in prime control. So, when we look into the lumen of the small intestine, if you could cut it in cross sections, so you see the circle, whether you see duodenum or whether you see Peyer's patches or nothing, you are going to see potentially these fin-like processes that extend from the wall of the intestine out toward the lumen. To me, they remind me of the fins inside your dryer. You know what I'm talking about? Those fins that stick out to help move the clothes around. All right? These are very similar to that, but these are called plicae circularis. And they're basically speed bumps. They're going to slow the movement of the chyme. The chyme kind of has to move around these things. But not only are they slowing down the chyme to make sure you've got time to absorb the material, you are drastically increasing the surface area. Now, these plicae circularis, you're going to have way more of them in the duodenum and less of them in the ileum because, you know, if you're absorbing in all three areas, I think you can imagine by the time you get to the ileum, you pretty much absorb most of the stuff. The ileum is just kind of mopping up. So the first increase in surface area are the plicae circularis. And so what we have on the surface is we have these projections up, these folds that are coming up. And the size of them, you know, in some areas of your intestine, they can be really tall and skinny, and others they can be shorter and more robust. But these finger-like projections in the mucosa are called villi. Now again, they get larger and more numerous in the jejunum. And why are we folding this up? Increasing surface area. Now, if you look in this illustration, you can see some blue dots, you can see some red dots, and they're different colors. I'm not sure what to call some of these, but some of them are kind of clear looking. So within each one of these villi, you're going to have an arteriole, you're going to have a venule, and you're going to have capillaries, but I want to point out this specialized capillary you're going to have from the lymphatic system. These are called the lacteals. Each one of these villi is going to have a lacteal. It's often called a central lacteal because if you look at a cross section, you're going to find them in the middle of one of these villi. and these lacteal are going to be there to absorb materials, especially fats. They're going to get absorbed into the lacteals. All right, so you've got these big fins coming out, the placae circularis. You have these folds of the mucosa on the surface of the entire luminal aspect of your intestine. Now, on every single columnar cell that you have in the mucosa, you're going to have these apical plasma membrane extensions. And these are called microvilli. The microvilli increasing the surface area because these are going to be the absorptive cells. Now, a long time ago in a land far, far away, early microscopists, when they looked at the surface of these intestinal cells, they said, you know what, these bristles on the surface look like a brush, the bristles of a brush. And so they call this a brush border. So the microvilli on the surface is a brush border by a different name. And what cellular biologists and molecular biologists have discovered since is that on these plasma membrane projections, you have some enzymes that are embedded into the membrane. And these enzymes are going to complete any chemical digestion that's not done, especially when you get into the ileum. But I can't stress enough, even with that, we are still absorbing material. All right, so here we have a villi. This is a cross section through a single villi. You see the columnar cells here? Each one of those columnar cells is going to have a lot of microvilli on the surface. Okay? Here we have our arteriole, we have our venule, we have our central lacteal right in the middle. All right? But what I want you to pay attention to now is the fact that when we come down the side, do you see how we go deep here? I mean, that's going to be our muscularis mucosa layer right here. And as we dive down into the lamina propria, these are going to be our intestinal glands. They're kind of like our gastric glands, but we don't have the pits. This whole invagination is just called an intestinal gland. And it's going to live between each one of these villi. You're going to have this gland. And so in the stomach, we produce gastric juice. Well, we're going to produce intestinal juice in our intestines. Hormones, a lot of enzymes, and mucus. And in fact, you can see in this illustration along our epithelial layer here, these columnar cells, what are those light spots? Any idea? Those little, in between the purple cells, those little white spots. They're called goblet cells. Somebody said goblet cells that are producing the mucus. As you get further down the intestinal tract, you're going to have more and more goblet cells until you'll finally get to a point it will look almost like it's just complete goblet cells, because the further you get down the intestinal tract, the more you're removing the water and you're making this hard feces, and you need more mucus for lubrication the further you get. So those are the goblet cells making the mucus. Well, there's goblet cells right there. Yeah. And they increase in number as you get toward the end. All right, so this is showing some of the cells that we have in our small intestine. We can see our simple columnar epithelium, and they're showing the microvilli. Do you see how that kind of looks like bristles at this magnification? Now do you see why somebody called that a brush border? So that's going to be our simple columnar absorptive cells. We've got our goblet cells into the mix. But then we get down into the intestinal glands, and this is where we are going to have cells that are secreting materials. Some of these cells synthesize gastrointestinal tract, peptidase. What is a peptide? It's a short protein. And we've got an ACE on the end. So in this case, enteropeptidase is going to activate pancreatic enzymes. And those are going to be dumped in at the duodenum. We also have some more of those enteroendocrine cells. And here in the intestine, this is where we're going to produce cholecystokinin and secretin. And what did cholecystokinin and secretin basically do?
They reduce the pH.
Shut down the stomach.
So wait, where they do in the small intestine?
So they're in the small intestine so that when you have material in the small intestine, that feedback goes back to the stomach to say, hey, we're down here in the ileum, we're telling you it's made it this far, you guys can stop working now because there's nothing in the stomach. And the baroreceptors and chemical receptors from the stomach are going to be doing the same thing. So multiple signals that are letting the brain know how we're doing in the processing of our material.
Multiple signals. Like
Yep.
check.
Hormone signals. You've got the enteric nervous system signals. Then you've also got the autonomic nervous system signals. They're all working together so that you know where the material is and how it's being processed.
Does it buffer pH too? Does it increase the pH?
No, we buffer the pH in the duodenum.
Okay.
And the pancreas may, I can't remember if it's got any secretions coming in that also do that. It's mostly those Brunner's glands in the duodenum.
Thank you.
We got more phases, but this one's pretty easy. Now we're going to move this material through the small intestine. And this is where we're going to use the muscularis layer. And early in the intestinal phase, we're going to do what's called segmentation, kind of kneading and chopping up the material. This is going to use that, principally the intercircular layer, just to kind of chop the material, move it back and forth. And these, of course, are going to be fastest in the duodenum where you're adding chemicals and you need to continue to mix these materials with the chyme. And as you move toward the ileum, where we're really finishing absorption primarily in the small intestine, you're not going to have the contractions as fast, this segmentation type. And it's why it's called early intestinal, because it's principally happening in the duodenum. Now, this later phase, the peristalsis, this is moving the material through. So this is going to involve primarily both of our layers, our intercircular and our outer longitudinal. Because we're going to have undigested waste, dead epithelial cells, the bacteria that we kill with the hydrochloric acid. We're moving all this stuff that we haven't absorbed and don't want to keep towards the colon. And so that's our late intestinal phase. The other was just kind of moving stuff around. This is moving it out toward the colon. Now we have another hormone that's going to help with this move from early to late. And this is going to be secreted by cells in the duodenum. And this hormone is called motilin. And this motilin is going to start, again, this moving the material down, which is called a migrating motility complex. Do you remember when we talked about peristalsis and threading the string back through the waistband of your sweatpants? Where you got the intercircular, the outer longitudinal. That's the complex action. You're working with both. So we're working with about 2 feet of intestine at a time to move material that 2 feet. Then we're going to have the next section take over, that next 2 feet and move, then that next 2 feet and move. It's almost like walking down the steps. And we're going to continue to do that until we get to the end of the ileum and we approach the colon. Okay, we made it to the colon. Wow. We've been processing this Big Mac for a long time. So, here we are going to move material past the ileocecal, ileocolic, either one of those names works here, sphincter, okay, and we know the sphincter is that enlargement of that muscularis layer. Normally this is going to be closed for business, but because of our signaling that happens and because of a reflex, we're going to allow the food, when it enters the stomach, that ileocecal valve relaxes, and that's going to cause the material, the chyme from the ileum, to move into the cecum. Now, we don't use that word a lot, right? Cecum. That is simply the blind-ended beginning of the large intestine, or the colon.
What do you mean by blind-ended?
You can only turn right. If you go left, it's a dead end.
Okay.
Well, hopefully we'll see a picture. Some of these pictures are not really up close enough. I think we'll see it in this one. Now, just like we saw with the pyloric sphincter, once that chyme moves into the cecum, the ileocecal valve closes to prevent regurgitation, backflow. So you open it, it passes, it closes, you can't go backwards. Because we're going to have peristalsis in the colon, and it's going to squeeze, and that material is going to follow the path of least resistance, and you don't want to squeeze the colon and it go backwards. Now, cholecystokinin, we're still playing here. It's not only feeding back to the stomach to shut the stomach down, but cholecystokinin is also, since it's a hormone, it's in the blood, right? It's going everywhere. Well, you have receptors in the ileocolic valve, and when cholecystokinin hits, that's one of the signals for that valve to open, to move material from the ileum into the colon.
So that just kind of chills everything out?
Yep. Cholecystokinin is the chill pill. Now, we need to get back here. We're kind of going back to the duodenum, because we're going to talk about accessory organs. And the first accessory we're going to talk about is referred to as the biliary apparatus. Biliary refers to bile. And the very first thing I want you to know and remember, because this is one of the things that most people miss on any kind of admissions test, where is bile produced? Bile is produced in the liver. Now, what organ do we often associate bile with? The gallbladder. The gallbladder is where it is stored, but it's produced in the liver. Okay? I do not want you to forget that. That's an easy one. So you produce bile in the liver. And from the right and left lobes of the liver, we have right and left hepatic ducts. Hepatic is a word for liver. And those come together to form the common bile duct. Now, when we get down to the bottom of the common bile duct, we have a number of valves down here. And if you're not eating, those valves are closed. And as the liver is producing this bile, and you see these arrows, they just go straight down the common bile duct. So bile from the liver is just collecting in the common bile duct. And it just gets more full, more full, more full, until it starts to overflow into the gallbladder.
Is that how you get gallstones?
Gallstones is when you have concentrated bile and bile salts. And when those salts concentrate, you can get a hard crystal, gallstones.
Because it doesn't empty out? Or?
Correct. So how many of you have had to add coolant to your car? Okay. Do you pour it in the radiator, or is there a little plastic reservoir that you usually pour it into? Yeah, it's that little plastic reservoir beside the radiator. This is like that little plastic overflow for your engine coolant. As your bile gets too full in all of these ducts, it just flows back over here into the gallbladder and is stored. And once you get that signal that you've eaten, especially a fatty meal, you're going to start contracting peristaltic contractions that are going to squeeze that little bulb of a gallbladder, and it's going to squeeze that bile and more bile down through the common bile duct into the duodenum. So the hepatopancreatic sphincter, that's controlling your pancreatic juice. You've also got, I guess this would be the, I don't know if it's called the cystic sphincter. I wish I could remember that name. I do remember the name of all of them collectively. It's called the sphincter of Oddi, O-D-D-I, because I think of Garfield's friend, Odie. Yeah, that's how I remember the sphincter of Oddi, or all of these collectively. All right, liver, we've already said the liver produces bile. You do have some bicarbonate, but primarily you've got bile salts. I don't understand the pigment part. I think the pigment part is going to be a waste byproduct. Potentially this could also be coming from the breakdown of the red blood cells in the heme, getting rid of it that way. You got cholesterol, mucus, all of that's going to help with your digestion. But principally your bile salts, this is what's going to help with the digestion of lipids. So bile and lipids is what I want you to think about here. We already said the gallbladder stores and releases the bile, but this is where when you talk of the gallstones, it also concentrates it. Have you ever done that experiment when you were in school? You get the solution of like salt or sugar water and you put the string in it, and then you let the water evaporate. As it concentrates, you crystallize those sugar or salt crystals on the string. That's how you're going to get gallstones if you're not careful. We've already said the cystic duct, that's going to connect the gallbladder that you see just a little bit of it down here in the hilum of the liver. That's going to connect it to the common bile duct. That sphincter valve, and I can't remember the specific name of this one, but the collection again is the sphincter of Oddi. That's going to control the release of bile into the duodenum. You do have a little sphincter, and I don't believe it's named, that controls the release of the bile from the gallbladder into the cystic duct. Man, I wish... Okay, I'm just going to go ahead and say it. I think it'll be coming up in a little bit. But bile salts, their principal function, so write this down somewhere. The principal function of bile salts is to emulsify fats.
It was on a previous slide.
I didn't see emulsification.
Yeah, right here, digest lipids, bile salts, and
But where's the word emulsification?
Digestive.
Yeah, emulsification. What does emulsification mean? Emulsification means you take a big glob of fat and you break it into a lot of small globs of fat. You've seen the dishwashing commercial where you got all the fatty stuff at the top surface of the dishwasher, and you drop a drop of that detergent in and it kind of all separates. And if you mix that up, all the drops of the fat will stay as small little drops instead of clumping together. The emulsification process creates more surface area of the fats for the lipases that are coming from the pancreas to actually work on to digest the fats. So I want to make sure we know that word, emulsification. And I think it's coming up in another slide, but I wish I'd put it on that one. All right, the pancreas. We know the pancreas has got two sets of materials. The endocrine portion of the pancreas, which we've already talked about with the endocrine system. Where do those cells live in the pancreas? The islets of Langerhans. But in this case, we have the exocrine, which means they come through a duct going right into the duodenum. We have the exocrine part that are going to be the pancreatic juices, and principally they're going to be all of these enzymes that these cells produce. Yeah, the acinus is just that round collection of glandular cells, glandular epithelium. There you can see a little illustration of one. And so some real creative person said, oh, let's call these cells of the acinus acinar cells. And really here in the pancreas, that's the only place you're going to find sort of this proper name for cells of an acinus. and that's in the pancreas, these acinar cells. So what are the digestive enzymes that are present in the pancreatic juice? Well, we're going to have pancreatic amylase, carbohydrates, right? Starches. Pancreatic lipase, what are we working on? Fats, triglycerides, lipids. We're also going to have proteases. What do proteases eat? Proteins. And in this case, we're going to have inactive proteases that are going to need to be activated downstream. Now, also coming from the pancreas, we got to have nucleases, right? Because you think about the materials we eat, whether it's plant material or whether it's animal material, they're going to be nucleic acids that we have to process. And so we get nucleases, and I think this is the first time we see nucleases coming from the pancreas. Now, it's a pretty good idea that we've got alkaline fluid, right? Because this is coming into the duodenum, and this is further going to allow us to buffer the acidic chyme that's coming from the stomach. And there's our bicarbonate ion. That's going to be an important part of causing that fluid to be more alkaline. Oh boy, here's our friend again, right? Cholecystokinin chills out the stomach, relaxes the ileocecal valve. Well, guess what? Cholecystokinin is also going to cause, when you have these fatty acids in the chyme, that's going to cause your peristaltic contraction of your gallbladder. So there's a lot that cholecystokinin does. And in fact, if a physician wants to know if your gallbladder function is adequate, they give you an injection of cholecystokinin. And if it hurts, then they're pretty sure you've got gallstones, or at least a gallbladder that's not working properly. Now, cholecystokinin comes from our enteroendocrine cells in the intestines. That's where they come from. And again, these are the things that cholecystokinin, we've already said quite a bit of this. The sphincter, I don't think we can add the ileocecal sphincter in here. But the gallbladder, you're going to release the bile, the stomach shuts down, the pancreas is secreting more of that pancreatic juice.